Okay, good morning. I'll ask everybody to start getting seated and again to settle in. We're going to begin with the program for today. So as everybody is sitting down, I'll go through the opening remarks here and make sure that everyone's had time to filter in before our actual speakers, the people that you want to hear from, get up here. So good morning and welcome to day 3 of Canola Week. This is the Canola Innovation Day. In 2003, alongside the canola industry meeting, Wilf Keller at the National Research Council organized a meeting focused on canola genomics to update this wider industry on the fast-evolving research field. But starting in 2012, Innovation Day expanded a little bit to cover topics that were broader than just genomics and inclusive of all novel, very interesting, highly relevant information to canola. So I'll be chairing this first session this morning. My name is Jed Christensen. I work with Bayer Crop Science based out of Winnipeg, but I'm very happy to be here today with everybody in the room. So we'll begin with a land acknowledgement. So as we gather, we acknowledge that we are on Treaty 6 territory and the homeland of the Saskatchewan Métis. Treaty 6 was signed on August 23rd in 1876 at Fort Carleton, which is 100 kilometers north of Saskatoon, and signed again on September 9th, 1876 at Fort Pitt, which is north of Lloydminster. The rights and responsibilities written into those prairie treaties did not end in the 1870s. We live those treaties today. On Canola Innovation Day, we think about the knowledge and creativity within all people living here on the prairies. We also think about our challenge to innovate in ways that improve canola productivity and profitability, but that also protect the land and the people that depend on it. Before we get rolling, I'd like to thank the sponsors, the platinum sponsors for this year's event, BASF, Corteva, and Bayer. Big thanks to our gold sponsors, AgWest Bio, Alberta Canola, Farm Credit Canada, Genome Alberta, Genome BC, Genome Canada, Genome Prairie, Ontario Genomics, Manitoba Canola Growers, NYA Ventures, and Sask Oilseeds. We could also not do this event without the support of Brett Young, Canadian Canola Growers Association, Cantera Seeds, Koch Agronomic Services, Syngenta, Blue Sun Scientific, Innovation Saskatchewan, MNP, and DL Seeds. This year, as I believe we did last year, we are using Slido to submit written questions, whether you're participating in person or listening online. Microphones are also available in the room. You can see a few standing here for questions at the end of each session. If you want to ask a question through Slido, for those in the room, on your table there is a card that says Canola Week at the top and has a QR code that will take you to the Slido website. And online, there should be details through the webinar chat function on QR codes and joining with Slido. For the organization of the questions and the digital aspect, Kati Navabi is running our session and helping as an online moderator. Let's see. If you need Wi-Fi in the room, there are details on connecting on the card. For those collecting CCA credits, we'll be showing QR codes throughout the afternoon and at the end of the day. For those joining us online, the CCA credit information will be put through the Zoom webinar chat. And in lieu of speaker gifts, once again this year, we're making a donation to Friendship Inn in Saskatoon. And so with that, we'll kick off the session for this morning. The first segment before the coffee break will be innovations in canola in breeding and genetics and diversity. And so we'll begin. I'm glad to introduce Axel Diederichsen, who is the curator and research scientist at the Canadian National Gene Bank for Plant Genetic Resources for Food and Agriculture, housed in the Saskatoon Agriculture and Agri-Food Research Centre. He'll be talking about changes to the access and benefit sharing regime for genetic resources from gene banks and how that affects plant breeding and research. And he's just recently back from a very large meeting on the topic in Lima, in Peru. So I'll turn over the stage here. There is, if you would rather, a handheld mic. Or if you have
I will speak here. Good. Thank you. Thank you for the introduction. Good morning, everybody. Yes, the title was read to you, Changes in the Access and Benefit Sharing Regime for Genetic Resources from Gene Banks Affecting Plant Breeding and Research. To start with, I like to show, since this is canola week, the 3 species that have canola quality oil and how much accessions there exist globally. 73,000 in gene banks all over the world. At PGRC, we have 2,184. And the right column shows how many of those we have, how many samples of those species we have supplied since 1998. It's more than 11,000 samples that went mostly to Canadian clients, several of them in the room here, but also abroad, because that is the task of a gene bank. And you see that I summarized also the other Brassicacea species, or Brassica species, that's the genus Brassica, so that includes cabbages and other more distant relatives that are also used in breeding. Now, what is behind all this is that we as a Canadian Gene Bank here, we take care of the plant genetic resources for food and agriculture, cultivated plants, crop, wild relatives. We have a collection of 120,000 seed samples. It's part of biodiversity, and biodiversity became a big topic with the Convention of Biological Diversity that was agreed upon in 1992. This is a legally binding instrument. And I can't even read how many countries. I don't have it. My glasses won't help there. But it's, you can probably read. Is it how many countries? 170? How many countries? I can't read it. Anyhow, doesn't matter. So the CBD came about. It's very big. Many member countries. Canada is one of them. The secretary is hosted in Canada. So Canada takes a lot of pride in this one. That had the effect when it came about, it emphasized the national sovereignty on genetic resources. Had the effect access became more difficult, and the plant breeding community, the research community noticed that, and they said, we are about food security. It's very important we find something to make sure that this enormous flow of genetic resources is preserved, and they established the International Treaty for Plant Genetic Resources for Food and Agriculture, which has a secretariat in Rome at the FAO, Food and Agriculture Organization of the United Nations. And these 2 agreements have 3 common objectives. One is the conservation of genetic diversity. The second is sustainable use of this genetic diversity. That includes the work by research and breeding. And the third is the fair and equitable benefit sharing from the use of those genetic resources. And that is a tricky part, the last one. What is important to notice is that the treaty has a multilateral system for access and benefit sharing as integral part of the treaty. It's legally binding. The benefit sharing aspect. It's grounded in the treaty itself. The CBD doesn't have that, but the CBD decided in Nagoya Protocol, which also does the benefit sharing, but always on bilateral level. And that's a big difference between the 2, and Canada didn't join the Nagoya Protocol. While in the treaty, it's legally binding and it's a multilateral system. How do I move? Did I move too far now? Can I go backwards? The red one. Okay. So access and benefit sharing. The treaty has this multilateral system for access and benefit sharing, the MLS. It covers 64 crops. The idea was to have all crops in it, but then when it came about, people start heckling. China said no soya beans, and then others said, oh, no soya beans, then we don't want this and that. So we ended up with this. In the end, China didn't ratify the treaty, so China is not part of it. But this is how it goes in international negotiations. We are now stuck with this list, it includes the Brassica complex that I wanted to mention here. It is, like all gene banks, pooled their genetic resources, access under the same conditions using the standard material transfer agreement from all gene banks globally. That regulates the benefit sharing. And the money that comes in from user-based payments, that was the idea, is also pooled globally and supports projects in third world countries, farmers that preserve a lot of diversity still, but also gene bank related projects. So the two things are pooled: the genetic resources, the access, and the benefit sharing. Why is this relevant for Canada? Here you see the 10 countries that request most of the MLS material of these 64 crops from global gene banks. This table is published, was published just now prior to the meeting in Lima of the governing body that happened last week. And there you see that Canada requested more than 200,000 samples from gene banks globally. What they did not show in this table is FAO in the report that 52%, more than half of it, came from our national gene bank. So we supply to the Canadian clients because we use this standard material transfer agreement also for all national clients. That's not done in all countries. The United States don't do it. They don't use the standard material transfer agreement for national clients. They use it when they ship outside or when they receive with it from other countries. They have to keep it. But for national clients, they don't do it while we do it. We make all of you signing to this. Therefore, we are also very serious in these negotiations to have possibly changes to the MLS. Here you see the distribution by our gene bank in general there, 152,000 samples since 2024. Since 2007, we use the standard material transfer agreement, and we have served 71 countries. Now the standard material transfer agreement, that is the benefit sharing regime where it comes down to money. There are two ways, or I should say, there are two things that are important. There are obligatory payment. If you use a genetic resource from any gene bank, you create a product, a new cultivar that incorporates material from the MLS, and you put restrictions on it. Restrictions means patents or the like. Restrictions means that others cannot use the material for research, breeding, or education without getting your permission or paying you a fee for it. If that's the case, as it is now, you are obliged to pay 1.1% of your sales minus 30% into the benefit sharing fund. For Canadian breeders who use the plant breeders' rights protections, plant breeders' rights allow anybody to use the material for research, breeding, and education. There's only a voluntary payment. That is how it is now. During the long negotiations that came to a conclusion last week, we had agreed to make the payments also mandatory for the second part, which was a major concession in the whole package we had considered. So I will tell you how it ended. This was presented to us as a little bit of background, and several of you come from the business fair, you will know better. Global seed sales, to give a little bit of figure, how much money is made from all these genetic resources, if you say that, right? 53.5 billion. That's a lot. That was in 1 year, 2023. 50% of those on GMO crops. That includes also canola, because most of the canolas have these are based on this GMO technology, at least in North America and some other countries. So that's amazing, the volume there, right? So what is also amazing, the top 10 companies already account for 70% of these this global income made from sales. So there's a high concentration, and genetically modified seeds are commercially very important. This is what the countries wanted to dip into this money. They say genetic resources are used. Why are we not getting a share of this? How does it look? Since 2007, the SMTA is used, and you see here's a table. They have the benefit sharing fund where I said the income is pooled, the payments are, and here you see who contributed to this benefit sharing fund that the treaty has. The MLS payments, which is user-based payments, since 2009, they collected the money. $36 million came in. Of those, 0.8 million, which is a little bit more than 2%, were user-based payments. And the idea was that these user-based payments should start flowing and create, support the work there. So it did not really work out as also we thought, probably, when it was negotiated. And we agreed we probably need to improve that. Therefore, as I said before, we conceded. Maybe also for plant breeders' rights protected material, we will have a payment obligatory there. Then you see the donations by countries, 80%. Several countries, in particular Norway, Australia, Italy, gave big amounts of money to keep this going. Every year, about $2 million went into this benefit sharing fund, but 80% were voluntary donations by countries. Then there are a little bit other sources. From Canada, the benefit sharing fund has received $3,187. That's not a million or what. It's $3,187. And it was a voluntary payment made by AAFC. I think the administrative costs behind were probably 3 times as high. Now, what will change after we had the big meeting in Lima last week? The governing body, which is when you have a treaty every 2 years, all parties, it's 154 countries come together, send representatives there. And in addition, we had an agreement was made in 2013. We need to improve the money flowing to the benefit sharing fund. 14 meetings of a working group with 24 representatives of 6 regions in the world, met 14 times with translation provided in the official FAO languages. Each meeting will have cost, I don't know how much, I don't want to estimate here, but that is what we did. And there were many informal meetings. I reached out to several of you who have received also emails requesting for input. No change. No change. We couldn't agree on an improved benefit sharing regime, and the reason behind was, for the most, this one: digital sequence information or genetic sequence data. Several countries have the wish that more money flows in, including us, but several say we must also make sure that anybody who uses information associated with the material has to pay, obligatory. In addition, a system to control the flow of all information that was generated of the material should also be installed to track and trace who makes use of the associated information. And that was, for us, from Canada, also from the US, that means from the North America region, and also for the Europeans, and for 2 countries from Asia, in particular Japan and South Korea, that was a red line. And we had said, when we resumed these negotiations that collapsed already in 2019, and they were resumed in 2022 at the governing body in India, we have said This is still a red line for us. We acknowledge that there is an issue with the information, and it's probably justified, but we say the treaty as it is set up based on the language is not the tool to really handle that. Therefore, we could have a voluntary thing, but not a mandatory thing regarding benefit sharing for information, and this tracking and tracing of information would not be acceptable to install under the treaty. That, in the end, resulted in a collapse. There were other things, but these were the key things. The Asia region, the Latin American countries, and Africa opposed strictly. So we had the North-South divide, and attempts to resolve it last minute, last week, failed. So we went home with no improvement. Other suggestions that were made, I mentioned already, the obligatory payments for all commercialized material. A subscription option was suggested also to enhance flow, so you pay right away. And a further monetary benefit for DSI, GSD was suggested, I said, but that was for us not acceptable. The information flow. And very important for us, an expansion beyond the 64 crops to create legal clarity for those who use crops outside. For example, soybean or flax, tomatoes, many vegetables are not in the present system. So what does this all mean? I think it's a very serious question, and we will have to think about it, even if there are no negotiations now, but the next governing body will happen, and the issue is still in the room. But a question really is also, how much are you or your company or your organization ready to pay for PGRFA accessed from the MLS? I showed you there's very little money flew back. I think that's a very serious question you should consider, because we still think user-based payments are the right way, not a general tax. Some countries do it with a tax, for example, Norway. How should a material transfer agreement look? Would the subscription be accessible? Do you need the other access options? I won't go into details there. And what about the benefit sharing for the digital sequence information, genetic sequence data there? Is it justified? And if so, how could that happen there? As a message here from all this, the access to genetic resources globally is getting more difficult, and maybe even after this meeting might be more. People may close and say, we don't have benefit sharing for DSI, we use our sovereign rights on the genetic resources, won't share it anymore. And many people don't share it anyhow there, despite being parties to the treaties there. So I think the access to plant genetic resources may become more difficult, and from a national perspective, it's probably a good thing to have a gene bank with material that you wouldn't easily access today and take care of it. So I close with this. Regarding this benefit sharing or the SMTA, we have a big construction site there. We also have a big construction site here in Saskatoon. Our gene bank gets an expansion of the cold seed storage. We need it. This picture on the left is from yesterday. It's in front of our building. The objective to have to the picture on the right there. So we are not yet done with all this. We have a break to think more, but eventually we need to resolve the open questions. Thank you for your attention.
Okay, good. Now we should be picked up online.
Okay, the question on Slido is the position of we see changes of farmers growing heritage varieties a valid method for in situ biodiversity preservation in Canada?
Could you please repeat? I didn't understand it fully.
The position of we see change.
Who is that?
It sounds
Seed
like
change. Okay.
We
I
see.
can elaborate.
Okay.
So Dana Maxwell, AgQuest, I'm also the chair for the Prior Recommending Committee for Wheat, Rye, and Triticale. With the policy recommendations that came out in the Seed Regulatory Modernization report from CFIA, one of the requests or one of the recommendations was around heritage and heirloom varieties and wanting registration on those. And I held a meeting with Weed Seed Change because they were the organization that were pushing that. And they were trying to claim that using those heritage and heirloom varieties in farms, largely organic, but possibly not exclusively, was in situ preservation of genetic diversity. And I'm wondering what your opinion is on that in terms of, is that a valid position? Is that valid preservation of diversity?
From a technical perspective, it's correct. If diversity is preserved on a farm and they preserve heritage cultivars, we would consider that as part of in situ conservation. More specifically, we distinguish there between crop wild relatives, which are in wild areas, that is also in situ conservation. In this case, it would be on-farm conservation as a specific type of in situ conservation. Technically, that is correct. Regarding the regulatory aspects, I know there are discussions ongoing, and there are very, how shall I say, intense discussions were ongoing last week also in Lima, because there's another work stream on farmers' rights in the international treaty. And there, Canada takes a position which is also enshrined in treaty language that this is up to the national authorities of each country to regulate that. It cannot be the treaty or activities that come from these works from cannot override national regulations. So it will be with the national regulators to figure out the details. I cannot respond on the details where this is or where this is going. I think the discussion is also not yet closed. I follow that with interest, but it's beyond me to comment on that. Thank you.
Okay, are there any other questions from the room? Last chances. Sally?
Axel, you said that I think it was Norway has a tax-based system. Where is that tax applied?
It was a demand. You know, these for the payments, also for the two payment options that I said to you, there are percentages applied. And we discussed a subscription system as one option that as soon as you request, you will pay every year amount of all your seed sales, a percentage goes into the benefit sharing fund. The rates suggested diverged by factors of 100 between the different groups. And the CBD has rates. The CBD created a system for the DSI, which is voluntary based on a resolution. The rates are also by a factor 100 higher than the top mark we had as a red line for the treaty. So Norway pays 0.1% of all seed sales in the country. The country has decided to measure that and pay. The country pays into the benefit sharing fund. And they say that would work. And many countries say all countries should do that. You saw the big volume of 50.2 billion, 0.1 percent of that you can calculate how much this is, 50 million or whatever a year. We have said we cannot come home and impose a general tax on all Canadians. We still think it should be a user-based thing, that those that utilize the genetic resources should be paying for that. Of course, that is, you could argue differently, but we say it should still be user-based. In the end, it will be then a cost that goes into the varieties, right? The breeders will need to recuperate the cost. In the end, it will be also something then that the producers or the farmers that buy these varieties pay in a way. But we still say it should come out of this system and not be a general tax.
Thank you, Axel. We'll move on in the program now. We have a pre-recorded presentation from Lauren Comin, who is the Policy Director with Seeds Canada. She couldn't be here today because she's at the GROW meetings right now, the Seeds Canada, I think. But she did present us with a pre-recorded presentation here, so unfortunately there won't be opportunity for questions following this, but we'll move into that now.
Hi everyone, my name is Lauren Comin. I'm Director of Policy for Seeds Canada. We're the National Industry Association for the seed sector from coast to coast. Want to apologize that I can't be there in person, but I'm very thankful that I was given the opportunity to give a very quick overview from a seed industry perspective on what the International Treaty on Plant Genetic Resources for Food and Agriculture really means to industry. So first, a little bit about the treaty. And I want to preface this by saying there are many interconnected treaties and agreements and discussions going on in this space. And they are not always easy to follow if you are not intimately involved in those discussions. I've been a part of this conversation for about 3 years, and I have to admit that I do not always have it straight, and I have to phone a friend many times just to understand what are the practical implications for the seed sector. So if you hear what I have to say today and walk away saying, I'm still not sure, it's okay. There are many resources out there, and there are many really good people who are on seed industry and industry side working on these negotiations. So the international treaty is designed specifically for plant breeding, and it facilitates access to genetic resources and material. So, plant lines, germplasm, etc. There are 154 contracting parties, and Canada is one of them. And the Food and Agriculture Organization, FAO, is the secretariat of the treaty. The treaty serves two purposes. The first is conservation of biodiversity. So the goal is to make sure that we do have long-term food security, and we are able to promote farmer rights to ensure conservation of genetic resources. The second is to facilitate access to genetic resources, making sure that there's a fair and equitable sharing of benefits of genetic resources, and to establish a multilateral system for access. So the goal of the treaty overall is that breeders are incorporating these diverse genetic materials into their breeding programs to ensure that there is continued biodiversity. So the scope of the treaty is all plant genetic resources for food and agriculture under the management and control of the parties. Canada is a party, and in the public domain. So this is only ex situ material. What that means is it's not material that's in its natural habitat. It's usually stored in a gene bank or some sort of plant library. For Canada, our materials are in the Plant Gene Resources of Canada, PGRC. There is PGRC in Saskatoon, Saskatchewan, and it is curated by Dr. Axel Diederichsen, who I believe will also be speaking to you and hopefully giving you some updates from his recent meetings in Peru. In the Canadian PGRC, there are more than 5,000 germplasm samples, including seeds, tubers, and cuttings. So every country who is a party to the negotiation, and even those who are not a party, will have some sort of a bank, and it may be a formal bank like we have with the Plant Gene Resources, or what you would think of as the seed vault up in Svalbard. But it could also be in universities and zoos, etc. There's also international institutions that will also have seed banks. So what is in scope in the treaty? Not all crops are included in the treaty. There are 64 species that are listed in Annex 1 of PGRFA. And those include the brassica complex canola. So that's relevant for all of you at the meeting today. And there's a number of other species in the brassica complex that are included. The multilateral system provides a standardized material transfer agreement, or SMTA. And it's standardized in a predictable contract, and it facilitates breeder access to the PGRFA. So that is one benefit of the treaty, is that it does provide a standardized contract for access. In theory, this standardized agreement should simplify the process, because it is standard across all parties of the treaty, and it avoids the need for countries to negotiate country by country bilaterally. So I want to get to the big so what. As I said, there's a lot of noise, and a lot of the noise you can sort of cut out. But what does it mean to your breeding program as a private breeder of canola, or as a public breeder, or someone who's working in germplasm development? So does it matter? It could, maybe, is sort of the gray answer here. If you don't access plant genetic resources, and you've never accessed from a national gene bank, either in Canada or another country, and you have no intention of accessing, then again, all noise, it probably won't matter to you. If you do access germplasm from gene banks, or you plan to access, then yes, it could matter to you. And what are the proportions of plant genetic resources from gene banks and incorporated into programs? Estimates from the U.S. suggest that about it's around somewhere just under 5% of genetic materials in commercial lines are from these plant banks. And I apologize, my image is right over some of the text, but no matter where you put it, it's going to be a problem. So what is being decided by the treaty? So one of the big pieces that private industry and the international seed sector has been paying attention to is how those who are actually accessing the plant genetic resources are going to pay for those resources. And this is under discussion right now as I'm recording this in a big meeting in Lima, Peru, which Dr. Diederichsen will be at. So if this has been decided, hopefully he will be able to present that update in his talk. But there's been a few different proposals, and the first is a subscription mechanism. So if you're going to be accessing genetic resources, you will take out a subscription, and it'll be a 10-year renewable term, and then you will pay for access based on a percentage of your sales. And right now, with discussions, you can exclude up to 2 crops from those sales. The other mechanism is a single access mechanism. So you will pay for what you access, and you'll pay a percentage of the sales of what you access for 25 years. So what are the next steps? The seed industry has been very actively involved in treaty discussions, as I mentioned. Seeds Canada is a member of the International Seed Federation, and they are involved. There are people who really understand this material who are involved in the discussions. The position of the seed sector has been, if you want to entice the private sector to incorporate plant genetic resources, the model has to be reasonable. There has to be a time limit or exhaustion of obligation. There needs to be flexible options for payment. If there is only a subscription mechanism, for instance, and it's a 10-year subscription, and you only want to access materials one time, that may not seem reasonable, and it may discourage you from actually incorporating those diverse materials into the treaty. And as I mentioned earlier, one of the goals of the treaty is to entice you to add resources into your breeding lines. Revenue must also be used appropriately, and that comes to the benefit sharing piece. Right now, it's kind of murky where that money is actually going to go and how it's going to be used. The Canadian government is also participating, and they are taking their positions from both private breeders and public breeders. So, Axel is in constant conversation with breeders across Canada, with myself and others, to make sure that the positions Canada is bringing forward are informed by industry. And Canada is seen as a rational voice at the table. So, rest assured, Axel and his team are bringing forward very common sense points. But ultimately, the decisions are going to be made by all the contracting partners. So if Canada isn't able to be allowed enough waste, or we're overruled, we could end up with something unfavorable. So, thank you very much. I know I kind of left that in a big question mark, suspense, but I'm hoping that Axel will help to fill in some of those, the blanks. And, the conversation isn't over. So, we'll keep seeing updates as the treaty continues to be negotiated. But again, rest assured, there are people from the seed sector, both in Canada and internationally, that are paying attention to the conversation, and will make sure that the sector understands, once things are decided, what the practical implications are to your business and your breeding program. You can reach me anytime, by email, by phone. Find me on the Seeds Canada website. Happy to connect and answer any questions you have resulting from this presentation. Thanks.
Good, and realizing that we should have switched the order of those presentations to heighten the suspense, and that we had spoilers. As Axel mentioned, unfortunately, the negotiations failed to come to a resolution. I will very briefly here, as part of those meetings in Lima, Peru, Bayer Crop Science did have a representative there, and I received a message from her this morning, and so I can share a couple of points. She also, Dr. Jasmina Muminovic, who was present there representing us, also notes that the negotiations failed to come to a satisfactory resolution, that the proposals for the standard material transfer agreement and benefit sharing through the multilateral system were seen as rather favorable to the seed industry, but there was a lot of concern expressed that the last steps of the negotiation were rushed, and that more time was needed to negotiate. And the main sticking points, I think, as Axel noted, were questions about the expansion of Annex 1 to include more species, and questions around the way that benefits could be shared for digital sequence information. As an outcome, the current SMTA still discourages our use of germplasm bank material. As Lauren noted, the obligations for payment are perpetual. So 30, 40 years down the road, even if gene bank material was used 17 generations back in your breeding program, you are still obligated to pay, and that is a challenge for us to sign on to. Yeah. So with that, we'll pivot from accessing diverse material and still hoping for improvements in the ways that we can do that more freely. And we'll shift into some topics about what we can actually do with diverse material, what we can learn from genetic diversity as we explore it. So our first speaker on this kind of pivoted section will be Dr. Steve Robinson, who is a research scientist with Agriculture and Agri-Food Canada. Joined AAFC approximately 20 years ago after coming from the John Innes Centre in Norwich in the UK, and has been studying brassica genetics and diversity for quite some time. Yes, so welcome to the stage.
Thanks for the introduction there, Jed. You're sort of betraying my age. So I'd like to start by thanking the organizers for the opportunity to present some of our work today. And we'll describe what we're doing at Ag Canada to support canola improvement. And so when I was invited, I was given the task to describe the plant material and the tools and the resources that we bring to bear towards canola improvement. But I also want to mention that perhaps the best resource that Ag Canada brings to bear for you guys are our staff. And it's the people that work at Ag Canada that really is the best resource that we have. And so I have the great privilege of working with a fantastic team of staff in the lab, and it's their ability to turn the ideas that I sketch out on paper and turn it into reality. And so without them, we don't get very far. And so I'd like you to bear that in mind. And so the work that I'm going to discuss today has been essentially completed by one person in the lab. And so that person is a highly talented geneticist by the name of Kyla Horner. And so it's down to her determination and dedication that the work I'm going to describe today is actually come to fruition. And so when I say we during the presentation, what I really mean is she. And so I've entitled the talk, Building Bridges to Success, and I hope that makes sense as we go through the presentation. Okay, so what is the nature of the problem? The problem is the natural history of Brassica napus. And so, as we all know in the audience here, Brassica napus is formed by an interspecific hybridization event between its diploid parents, Brassica oleracea and Brassica rapa, contributing their A and C genomes. As soon as this happens, the consequences are that Brassica napus becomes genetically isolated from its parents and all of the wild relatives. And so this provides a profound bottleneck and a lack of access to alleles which are valuable and that we might want to use in the future. And this is not unique to canola. It affects all of our polyploid crops. And so all of the diversity that you might want to access is in the crop wild relatives and in the diploids in the case of the brassicas. When you look in napus, you have the domestication process and you get further erosion of the diversity that's available through breeding and the development of elite cultivars. And so over time, we're basically throttling our ability to develop and address the challenges that we've heard about in the last couple of days. And so this is really what we're trying to think about to address. And so that's not to say that there's no diversity in Brassica napus, and so we have a successful seed industry, and they're able to use a wealth of approaches to develop new cultivars, which they make available to you all. And so the process becomes very integrated, and you end up with a bewildering array of data to support these variety developments. And so at Ag Canada, Where I think our lab can make the best contribution is at the beginning of the process, where we can generate new diversity as a feedstock for these, to support future variety developments. And part of the success that the public sector's had is in developing new genomics tools and sequencing the genomes of these crops and developing SNP arrays where you can quickly genotype all of the diversity that's available. And when you do this, what you learn is that the great deal of diversity that you find within brassica napus can be explained by the different varieties which have different growth habits. And so you end up with the diversity being separated into winter, semi-winter, and spring growth habits. And so then the diversity within each of those gene pools is relatively limited. And of course, in Canada, we are focusing on the spring material. When you dive down deeper and you look to see where the variation is in brassica napus, you find that the problem child is the C genome. And the C genomes are essentially the same across all of the brassica napus, as far as I'm concerned. The diversity is very poor. And so then we can define these are the two problems that we need to address. We need to find new diversity, sources of new diversity that we can introduce, and we need to develop new methods to access that diversity and deploy it in a sensible way. Okay, so we've heard in the last two talks, over the last 50, 60 years, there's been efforts to conserve germplasm for crops and crop-wild relatives, and these are stored in an array of gene banks, and these are essentially gold mines that we can dig, find useful variation, and bring it and make it available to canola breeders. And so in Saskatoon, we're very lucky. We have the PGRC on site, and so I can stroll down the hallway and talk to Axel and interrogate the databases, and I can sign his forms and get access to seed quite readily. And so in that way, we can solve problem one of finding access to new variation. So then the question is, how do we solve problem two and develop methods to introduce this variation into the gene pools? And so the time-honored approach, which is adopted by many geneticists, is to take the diploids that you're interested in, form a cross between Oleracea and Rapa. Oleracea donates a set of C genome chromosomes. Rapa will donate a set of A genome chromosomes to form a sort of amphihaploid. And then you can double these chromosomes using drugs like colchicine to form a new synthetic tetraploid. And this works, it's rapid and it's relatively successful. But the problem is that you get exactly the traits that you don't want. And so these new synthetic napus are self-incompatible. They set poor seeds, they're often sterile. They are late flowering, they require vernalization, and they have essentially poor vigor. And so to use them, you've then got to do a lot of work down the line by crossing them to existing napus lines. And you essentially erode the very variation that you're trying to integrate. And so the question is, can we do better? And so, as I mentioned, there's something special about brassica napus. And so brassica napus is self-compatible. It sets copious amounts of seed, and we can adapt it to the Canadian environment by making it early flowering. It doesn't require vernalization, and it can have good growth vigor. And so something happened back in the day by breeders in antiquity where they selected for alleles which we can no longer access when we make these crosses between the diploids. And so the question then becomes, can we exploit this? And when did this happen? And so it's been estimated that this happened 3,000-4,000 years ago in the Mediterranean region. And so we can speculate that the Romans might have been in charge of this. And so it might have happened in a Roman garden. Probably the crosses were made by a slave, perhaps a female slave. And so we should be grateful to that person or people, for they are the people that have given us canola. And so then it occurred to me that we can exploit this, and rather than being hasty and making crosses right away between the diploids, can we first take our time and domesticate the diploid gene pools? And so can we take the things that make canola special, put them into the diploid gene pools, and then only then turn around and make resynthesized Brassica napus? And in that way, if we can do that, we can solve the problem of self-incompatibility, solve the problem of poor seed set, and if we can do that, can we also make them adapt to the Canadian environment by having no vernalization requirements, having an early flowering phenotype with good growth figure? And if we can do that and have that, establish that as a package, we can then pass that on to the breeders and they can finesse them, combine them with good agronomy, the disease resistance they need, and then we can release new varieties. And so in a way, what I'm trying to say is, can we rethink what Brassica napus is? And so that's what we think about in the lab. And so you have to imagine a scenario where we might be able to do this. And so what you'd end up needing to do is to develop a new species or bridging line, which represents access to the diploid gene pools in Oleracea and Rapa. And then if you can do this, you can make crosses to the array of diversity which is available at the PGRC, including the wild relatives. And so you really can think about reimagining what Brassica napus is, and you can potentially increase the heterotic potential in this new material. So then, what is the problem? Well, the problem is these bridging lines, they don't exist. So how do we solve this? So what we could do is we can take Brassica napus, and we can make a cross, as I mentioned, with Brassica rapa on one side, Brassica oleracea on the other side, and you end up with triploid material. And so you essentially end up with a set of aneuploid chromosomes belonging to, which would have originated from Brassica napus. These lines are going to be difficult to work with because they're going to be largely sterile. But if you can rescue the embryos, work with seed set, you can make crosses, back crosses essentially, back to the parental diploid material. And in that way, the aneuploid chromosomes will start to segregate. And by taking a recurrent backcrossing approach, you can eliminate these aneuploid chromosomes and return to a karyotype in which I will have 18 chromosomes on the oleracea side and 20 chromosomes on the rapa side. But if you make selections for the alleles that originate in napus, these alleles which these breeders in antiquity selected for, you can move them into the gene pools. And so that's what we're attempting. So is this a realistic strategy or not? And so you can do some sort of calculations. We know that we're going to be dealing with small population sizes because this material is sterile and you've got to rescue every single embryo that you get from this material. And so if we're going to manipulate a number of traits, and so we want to manipulate seed set, self-compatibility, early flowering, vernalization, vigor, so 5 traits, you can do the math and calculate the population size that you'll need, and it becomes rapidly apparent that you can really only manipulate 1 or 2 of these traits at once. And so we need to figure out a strategy where we focus on the most important alleles for domestication from Brassica napus and then try to make the adaptions that are required to adapt the material to the Canadian environment by using diploid material. And so that's the strategy that we use. And so just to summarize, it's a strange concept, but you have the Brassica napus genome, like a nucleus of Brassica napus containing those A and C genome chromosomes in one nucleus. And so what we're trying to do then is make crosses to move these alleles, which are in napus, into the A genome on the left and the C genome on the right, and using markers, we're going to select for the recombination events that occur during that breeding process. And so in that way, once we've got this material, we can manipulate it more simply. And so we're going to focus on the self-compatibility alleles for napus and the fertility alleles in napus, and we're going to move those alleles into the diploid parents. And so what Kyla did was, In order to do this, you've got to find the right material in the diploid lines, and so we had access to a double haploid population of Oleracea derived from a cross between TO1000 and early big. And so if anyone knows these material, TO1000 does not require vernalization. It flowers early, but it's like a big Arabidopsis, and it's agronomically very poor. Early big is a broccoli type of plant. It's robust, it's strong, it's got a thick stem. Agronomically, it looks nice, but it flowers late. But we have the segregating population, and so what she did was she characterized the entire population, phenotyped it for a range of different traits, scored them all, including seedling vigor, where she was measuring the rates of development at low and high temperatures. And so then she selected the line which had the sort of aggregate best ability to deliver what we need. And so she landed on line 34. Okay, so that's what we're going to use as the parent in the C genome side. And so just for brevity, I'll focus on what we did, the strategy for the C genome side. We are doing the same thing on the A genome side, but the C genome side is the real problem, and this is where the barriers are profound, and it's the reason why there's low diversity on the C genome side of Brassica napus. And so she made a cross between Brassica napus using the topaz genotype and TODH34. She forms an F1, and so this is a tetraploid material. She then backcrosses it back to TO1000 to create a B1 generation where it's going to be segregating for these aneuploid A genome chromosomes. And you'd expect to lose half of the chromosomes as they segregate to the poles during meiosis. So you'd go down to five aneuploid chromosomes in generation 1. In generation 2, you'd lose another half of them, so perhaps we'd get an average of 3 aneuploid chromosomes. Back cross 3 would get us down to 2 or 1 chromosome if you're lucky. And then back cross N, we need to get rid of all of these aneuploid chromosomes to return to the status of the Brassica oleracea karyotype. Okay, that's the plan. I cannot emphasize how hard and the dedication that she's done to achieve this, and involves crossing and rescuing the embryos and tissue culture and encouraging these plants to survive, and then genotyping and making selections for the right recombinants. And so she's dealing with very small populations of extremely valuable material. And so what I asked her to do is extremely difficult. And on top of that, we had a global COVID pandemic where we shut the building down and made it almost impossible. And so, honestly, what she's achieved is honestly astounding. And so, just to detail some of the material, the way that she did it, so she's made like 1,500 crosses. She's recovered over 100 embryos per generation. 42 of these plants will survive, and so you can then genotype, make the selections for the C genome alleles that we want, for self-incompatibility, or the self-compatible alleles from napus, and fertility alleles from napus, and then you get your aneuploid chromosomes, and so you get an average of 6 aneuploid chromosomes in generation 1. Again, she makes over 1,000 crosses, rescues all those embryos, encourages those plants to survive. You get aneuploidy, we're down to, on average, aneuploidy of 4 A genome chromosomes. Generation 3, again, over 1,000 crosses, lots of embryos, plants to survive, and so we're pushing these down and we're making selections. But at each generation, you can't select for the least number of aneuploid chromosomes because you're constrained by selecting those individuals that possess the topaz alleles that you need, which confer the self-compatibility and the potential to recover fertile. You'll end up with material which will set seed in the future. So we go through a backcross 4, and so, it's like, is this going to work? And, you're struggling, you're on the edge, but she's recovering plants, so you've got 25 plants which survive, and eventually you get to generation bridge, where we've got rid of all of the aneuploid chromosomes. We've selected for the alleles that we want from topaz on C6 and C7, and we can then use this for crossing to access the wider wild diversity which is housed at PGRC. And so we collected this material, we get access from Axel. We need to genotype it, so we developed a technology we had a capture array where we can sequence the diversity that we've been provided. So that includes oleracea and a bunch of wild relatives. And then so by selecting individuals at the edges of this PCA graph, you can select the lines with the greatest diversity. And so this is what we do. And so although we get most of the diversity is in oleracea, we can use material from Encana or Velosa or Montana on the seed genome side. But in that case, we're having to manage vernalization requirements as well. And so this is the stage that we're at, and so we're expanding. So we're essentially domesticating these wild relatives by transferring the Napus alleles through this bridging line into the wild relatives. And then the idea will be that we can then combine them with material on the A genome side, and we'll complement them and we'll bring them together to form new Napus. And so we can then rethink what Napus will look like, and it can look profoundly different. And so, again, when you make these crosses, 50% of the genome will be from the bridging line, and if that's not good enough, you can make back crosses to increase the diversity of the line before you make these synthetics. And so there's lots of different strategies that we can think about to increase the diversity as we go ahead, but it will mean managing vernalization as you go along. So how do we use this resource? Well, we've heard the call to arms from the community here, of the traits that they need. And so one way we do non-targeted phenotyping. And so we can go and use what natural selection has allowed, where we can collect material which has evolved in regions where the stresses are apparent. And so in these regions, natural selection will have increased the frequency of the alleles that we want in the material, and so we can access that. And so this can be found at PGRC, and Axel is happiest when he's showing off his plots. And so I go out to the field with him, and this year we were looking at his brassica 240 lines, and so these can be integrated as part of the A genome side of things, and so this is wonderful to hear. We can do things more targeted, and so again, these can be driven by people in this room who set priorities for disease resistance, and we collaborate with Hussein Borhan, and so he provides us with material which has got disease resistance, and so then we can integrate them using this strategy to bring them into the tetraploid gene pool. Other methods that we use, we've got a resource in Saskatoon where we've got this lysimeter plant array where we can do detailed drought experiments. And so if you're interested in abiotic stress, we can screen variation to find lines which better respond to drought recovery. And so you can use your imagination. Once we've got these tools in place, we can reimagine what Napus might look like and solve some of the problems that you're experiencing on your farms. And so I just want to summarize by bringing it together. We're at the stage where we are moving these alleles into the wild germplasm, and we have a couple of years left in this project, and by the end of the project, the intent is we will have generated new Napus, and we will be able to make that available to the community. And so I'm just a simple geneticist working at Agriculture Canada. My role is really to demonstrate what is possible and provide the tools for the larger community. And it's for the breeders, industry, producers, society as a whole to determine what their crops will look like. And I just want to sort of acknowledge those people involved. As I said, the driving force behind this is Kyler Horner. We're grateful to Dallas. He's the person that provided the material from Axel, and then I'm grateful to my colleagues, my long-suffering colleagues who put up with me and all of the nonsense that it involves. And of course, we're very grateful to the Canola Council of Canada for funding this, and the commodity organisations who are bold enough to fund this type of research. And if you'll just allow me indulge, I've been asked to sort of advertise an upcoming meeting here in Saskatoon, and so it's a save a date kind of thing. And with that, I thank you for your time.
Good. I think we'll take a little bit of time maybe for a couple of questions. There's one on the Slido.
Someone is asking out of curiosity how many back crossing to reach the bridging.
Well, in the case of the Oleraceous site, it took us 5 generations. But I mean, you can't predict this. You get what you get with your recombinations, and so we were anticipating it would take at least 4, but it took 5. So yeah.
You have time for one more?
Sure.
Hey, Steve, Jay Wetter. Sounds exciting. I don't really know what you're talking about mostly, but just wondering whether, in light of the conversation from yesterday, whether the private breeding companies are engaged in this work and whether they're as excited about it as you are.
Currently, they are not. I mean, I hope I've been able to communicate what we're doing. I would imagine that the geneticists and the breeders would be excited to access this material. We've been getting on with it rather than advertising what we're doing. The first time that we spoke about this was in the Brassica 2025 meeting in Gießen, and so now that we've broken the back and that we're confident it's going to work, that's when we're sort of advertising what we're doing. So now's the time to engage, I guess.
I'll just start by, I thought it was interesting that you feigned sensitivity to references to your age and yet kept references to Monty Python in your talk. My question is, you made it seem like the technical difficulty was the embryo rescue. Could you elaborate what that entails?
I mean, embryo rescue is challenging, but it's something that when I say we, I mean Kyla does it routinely in the lab, so it's not although it's challenging, it's something that we can overcome. The problem is the sterility, and so the chromosome imbalance and the balance between the ratios of the embryo and the endosperm causes like profound sterility in this material. And so before we embarked on this project, the sort of the law from the literature would sort of suggest that it would not work, and we'd be crazy to attempt it. But anyone that knows me knows that that's just a challenge. So yeah, we've essentially proved that you can do this, and we've done it. And with these tools, we really can rethink what Brassica napus will be in the future. So.
Hi, Steve. It was a very fascinating presentation. Thank you. I was wondering, you put up humongous work to get this done. Using gene editing technology, how it's feasible to get this achieved is much faster way.
Yeah. So, I mean, so you'd have to. So first of all, you'd have to transform. You'd have to identify the alleles that you wish to interrupt. And then you're assuming that you're going to knock them out, and then that would cause the trait that you're after. We don't know what these alleles are that cause the barriers to fertility. And so what we're doing is we're moving regions of the genome, which are essentially QTLs. And so in theory, if you knew the molecular biology and the biochemistry in detail, then yes, you could make those edits and do and achieve those things, but we're not at that stage, and so we have to do it using genetics, and so this really is a power as a demonstration of what you can achieve using old-fashioned, good old solid genetics. Yeah.
Thank you.
One online. Sorry.
Yeah.
We'll take. There's one question online, and then maybe we'll come back.
Yeah.
To yours, and then we should probably move on. Yeah.
Thank you. This one might be quick. Someone is asking, why not just introgress from Brassica oleracea to Canola? Why do you need to make synthetic lines?
Okay, so Brassica oleracea is a diploid, so it has 18 chromosomes, and Brassica napus is a tetraploid, it's 38 chromosomes. And so when you make that cross, it basically doesn't work. You have to work hard to recover what is essentially then a triploid, and the triploid is completely sterile. I mean, that's the problem. I mean, these barriers are profound, and so when we decided to do it, we had to commit, and we had to commit resources, and we're working with material which will not set seed. So there's no stopping. So when we started, we knew that we were taking on quite the endeavor. Yeah, so I mean, you have to make these bridging lines, and then you have to use these bridging lines, and then to address the complications of introducing all the alleles which make things adapted to the Canadian environment, that makes it even more tricky. And then when you shut the building down during COVID, that's more fun.
I would say that Steve, you've done a wonderful job. But my question is, did you use any molecular marker assisted selection?
Yeah,
particularly oleracea and the Agitopus closing. Yeah.
Yeah, of course. So yeah, I mean, absolutely. All of these QTLs, all of these regions are all tagged with molecular markers. We wouldn't be able to even imagine attempting this without all of the resources that have been established by researchers who are ahead of me down the line that have developed. And so we really are building on their shoulders. And so all of the SNP arrays and the genome sequences and the tools and the fact that the detailed mapping which has gone into determining these regions, we wouldn't be able to do without all of the efforts that have come before us. And so all we're doing is bringing them together to try to reimagine how we might use this tool in the future. And it wasn't me, it's all Kyla. So it's Kyla who deserves the credit. As far as I'm concerned, in my eyes, she stands next to these ancient plant breeders.
Very good. So thank you. Excellent presentation and very good engagement. Lots of excellent questions. We'll move on to the last presentation for this session. We're a little bit behind, but I'm sure everybody doesn't mind because, again, there's been such great engagement and questions here. So for the last presentation, we have Kamal Khadka, who is an assistant professor of plant breeding and canola breeder at the University of Idaho. And so they'll be joining us by virtual presentation that I believe we have queued up here. So with that, we'll turn over to the virtual presentation. I think, Kamal, are you on the line?
Yep.
Great. Go ahead and take it away then. I think I heard you say yes, you were on the line.
Yeah, can you hear me?
Yes, we can hear you now. So okay, feel free to launch into it.
I want to share my presentation. Okay. Can you see my screen now?
Yes, it's up on screen here. Yeah.
So it's in the presentation mode, right?
Looks good.
Okay, great. Okay, good morning, everyone. I'm Kamal Khadka from University of Idaho. It's still a little early here in Moscow to wake up and give a talk. Anyway, so today I'm going to talk about increasing efficiency of utilization of genetic resources from gene banks. My presentation is not going to be very technical. It's more about my thoughts how we may be able to improve the efficiency and utilization of genetic resources from gene banks. We all know what gene banks are, why they are there for. So and we have so many new technologies coming up to work on the genetic resources. That's all there. But in my presentation today, I'll be basically focusing on some approaches which may help improve the efficiency of utilization of these resources in gene banks. So I think it all starts from improving the gene bank operational activity itself. So I was listening to one of the podcasts with research leader of USDA ARS Plant Introduction and Testing Research Unit at Washington State University, Dr. Marilyn Warburton. So in the postcard, in the podcast, Dr. Warburton emphasized the importance of modernizing the gene banks, So in the podcast, Dr. Warburton also talked about the 2018 Farm Bill, which requested the scientists and researchers in National Plant Germplasm System to identify causes of backlogs in gene bank activities. So since then, the improvement have been started in gene bank operational activities. And basically, one of the major targets have been to establish or have the germplasm with better quality so that they can be better phenotyped and better genotypes. And in fact, the gene banks, most of the gene banks in the United States, they have been, they were established in the 1940s or 1950s. And in fact, and many of them need some improvement in their operational activities. So we have been, in the recent years, we have been hearing about gene bank phenomics. In fact, it's very important. It's a really important topic to talk about. So gene bank phenomics, basically, it's a strategic approach to enhance the value of genetic resources that are deposited in the gene banks. And as I said earlier, there are many high-throughput phenotyping platforms available for large-scale phenotypic characterization of the gene bank materials, such that there is high possibility that we can fulfill the role, their biorepository role, or the role for conservation and breeding. And combined with the genomics, that's it's really going to improve the value of the work that we do. Again, in the recent few years, we have been hearing about gene bank genomics. So one of the challenges with the gene banks that I understand is the lack of effective means for the discovery and transfer of beneficial alleles from gene bank resources into modern varieties. So what I think is that with the employment of genomic approaches, molecular passport data can be generated, which has potential to increase the efficiency of the plant breeding programs. And in fact, the employment of these high-throughput phenotyping and all this pre-breeding work, with all this, I think we will be able to efficiently exploit the gene bank resources. But in my presentation, I'm more talking about the general gene banks rather than focusing on the genetic resources associated with canola. So here in this slide, what I'm trying to show is that different CGI centers. They house gene banks, and what I have understood is that city centers like CIMIT or International Potato Centers, they have been investing quite a lot in pre-breeding activities. And here in this slide looks a little, clumsy, but still, what I'm trying to show with this slide is that, the genetic resources, the brassica genetic resources are available in different gene banks all over the world. I think there are more than 80,000 brassica the genetic resources of brassica germplasm available around the world. And if you look at the United States, we have more than 2,000 germplasm available for use. Some of the public institutions have been working on the pre-breeding part, and University of Idaho itself, I started the position just over 2 years ago, and I have also started working on pre-breeding of canola for the threats associated with acid tolerance. So there are other public breeding institutions who are also working on some pre-breeding work. So my concern is, are we efficient enough when we are working on pre-breeding, or can we become more efficient? That's another question. Based on my understanding, private sector breeding has been tremendously successful in for some of the crops. However, some questions come into my mind. So is private sector investing more on trait design, which could potentially be reduced? And another question, which may not be relevant in case of canola, but it comes up in my mind. So will the private sector be interested in other crops that are important for us? So that may not have the commercial value as some of the crops which the private sector have been successful. But the important thing to note here is that the public breeding program has been significantly investing significantly in pre-breeding kind of work. However, one question that comes there that is raised is, are the outputs from this pre-breeding work in public breeding programs work appropriately utilized? That's another question. So I will be connecting this one, this pre-breeding work with some slides that I'm going to talk about now here. So one of the challenges that I see with the public breeding programs is the funding. So Margaret Smith of Cornell University, in a keynote speech in 2017, indicated ensuring sustained funding for public breeding programs in the US is an ongoing challenge. So I was reading one of the recently published papers related to sustaining public plant breeding programs across generations. So in this paper, what has been indicated is that in the United States, more than 60% of the public plant breeders are in a process to retire. So one challenge that has been observed is the transition. So the knowledge as well as the germplasm transfer from one generation to other generation has not been that good. So with that, there is loss of knowledge, there is loss of germplasm as well. And I read another paper which talks about the plant breeding capacity in U.S. public institutions. So this paper is based on a survey study to understand the status, course and needs plant breeding program. Needs a plant breeding program. So there were altogether 278 public sector plant breeding programs from 44 states responding to the survey. So the result clearly showed that the many programs indicated budget shortfalls endangered the capacity to support personnel and operations. That means funding is one of the key challenges in the public breeding institutions in the United States. But the important thing to note here is public breeding programs have the capacity to do the pre-breeding kind of work. And I was reading some articles that showed the concern over public that showed that public plant breeding is neglected. So this one is this article is by Doug Miller, the executive director at Canadian Seed Growers Association. So in the article, it states that Canada's public plant breeding system, which is the foundational force, the silent insurance policy, and now seemingly the forgotten workforce of Canadian agriculture. That means it's basically associated with the wheat and barley breeding program. I'm not exactly sure how it looks like in case of canola, but in general, it seems that funding cut in the public program is going to hurt a lot of public breeders. And this is another article that I read, which shows concerns over reduced government funding on wheat and barley. So my worries as a public breeder is that I know that both public and we all know that both public and private sector breeding needs new generation of plant breeding scientists. And I understand that running academic programs, mentoring new researchers, it requires ample opportunities for hands-on experience on breeding research. That means it demands good funding, which seems challenging in the context that breeding is slowly moving away from public sector. But here, as I already said, the public breeding has some strength. So among the core plant breeding activities by the public breeding sector, like the universities, so based on the results from the paper that I talked about before, the paper that was published in 2020, the survey-based paper. So this indicates that variety development occupied about 33 or 34% of FTE, of total FTE. So, and pre-breeding activities occupied more than 65% of FTE. So this clearly indicates that plant breeding, public breeding has a strength in pre-breeding, which could be one of the major things, one of the major strengths that in the future we can use it for increasing the efficiency of utilization of genetic resources in gene bank. So with this, the question here is, can we establish a robust public and private partnership in pre-breeding gene bank genetic resources? So my thoughts on why public-private partnership may yield success. So gene banks themselves do not have full capacity for extensive pre-breeding work. Definitely, quite a lot of work is going on, But again, how much is the question, right? And pre-breeding, the important thing that we need to note is that pre-breeding requires a long-term commitment of funding resources. So especially in case of public breeding institutions, funding is a challenge right now, and looking at the future, it's going to be a long-term challenge. So what I'm thinking right now is the private sector may be able to contribute the resources, and they may save significant amount that they invest onto a design. And the other thing is that we do quite a lot of work using the checkup dollars, and my personal thought is it may not be wise to use the checkup dollars in pre-breeding activities. And I read one paper which was based on one international workshop in 2022 that happened that was in Netherlands. So this workshop was on transformative impact of genomic technology within gene banks. So all the participants, there were diverse participants from CG centers, participants from academics, participants from private sector. So the stakeholders, they all agreed that that public-private partnership is critical for utilization of genetic resources. So they clearly indicated that public-private partnership is crucial for efficient utilization of genetic resources. And I was reading one of the a little old blog. So the blog was on successful story of public-private partnership in canola. So in this blog highlights the significance of public-private partnership, where Dr. Wolf Keller shares his experience to bring genetically modified herbicide-tolerant canola from labs to farmers' field during 1985 to 1995. So he says that for canola, without public-private partnerships, the development of genetically modified varieties wouldn't have happened. It makes sense to partner. And I saw another example in a successful example of successful collaboration in canola research. So that is between the NPG, a German-based breeding company, and University of Western Australia. So canola pre-breeding at University of Western Australia has been supported by NPG for more than 20 years now. So this partnership has resulted in more than 37 valuable commercial canola cultivars for Australian farmers. So with this, what I see is that there is possibility to possibility of partnership between the public breeding institutions and the private sector for improving the use of genetic resources in gene banks. Definitely, with partnering with the private sectors, there are going to be some challenges associated with benefit sharing and the intellectual property right. But since we all have the common goal to improve the efficiency of utilization of genetic resources, be it a public or private sector, my feeling is that there should be some ground for appropriate agreements for partnerships. And another thought that I have is that phenotyping or pre-breeding gene bank genetic resources should be the least disputable work for public-private partnerships. And in fact, it needs the trust between the institutions that are partnering. So that's all from me. Thank you so much for your patience. If you have any questions, I'm happy to take the questions.
Thank you very much. As you noted, there's an opportunity for questions. I don't know if we have any online. We do have one. Okay.
What are the major funding sources that are supporting your breeding program, and what percentage from public sectors?
For me, since I just started my position over 2 years ago, I have been getting the support from the commissions, Idaho Oil Seed Commission and Washington Oil Seed Commission. And apart from that, I have one USDA NIFA grant. Basically, those are the ones. Those are the only supports that I'm getting right now.
Good. Are there any other questions in the room? Any other questions online? I think we're okay. That's good. Okay. Well, with that, we are a little bit over time, so we will wrap it up. But I would like very much to thank all of the speakers this morning. Again, excellent presentations, very good engagement and questions as well. So one more round of applause perhaps for the speakers. Yeah. Good. And right before we break, I'd like to invite up Chris Mancher with the Canola Council to make some presentations of the awards for the poster and flash talk competitions.
Okay, good morning, everyone. Just want to thank all of the poster and flash talk presenters for their presentations, as well as the posters that were found just in the break room over there. Our subcommittee had some difficult challenges because there was actually a lot that were rated very highly. So I want to commend everyone for having really good presentations and also the quality. And hopefully there was a lot of interactions between all of our members here at Canola Week, and that'll hopefully stimulate some more discussions in the future. So we're going to start off with just the best poster, and then we'll go into the best flash talk. And I'd like to announce Virginia Miguel from Queen's University for the poster titled From Arabidopsis to Canola, CPK 28 Homologs in Pathogen Resistance. And also for the best flash talk, Mohammed Mukhtar from the University of Manitoba for his flash talk titled An Applied Approach to Verticillium Stripe Management in Manitoba, a Proof of Concept Study. So hopefully I see Virginia here, and hopefully Mohammed's also nearby as well. And at the break, we can take some pictures, as well as provide the certificates, as well as a nice little gift card for them for recognition. So thank you very much.
Okay, great. We will wrap up for a coffee break now and try and get back seated and start the next session at 10:40, which is a bit shorter than in the program, but we'll try and get back on track. Thank you.